Hello, good afternoon, everyone. Let's get started. We are going to be finished, not finished, but like we'll go through a majority of lecture 4 today. And I will also want to explain to you what the lab is about. As you know, the lab is going to happen next week, and you will have to submit it next Thursday. All right, so I'll take a moment, maybe halfway through the lesson, to explain what needs to be done and where to find the information and so on and so forth. But let's begin with um, the lesson. So last time we were talking about the structure of the cell membrane, and we know the cell membrane is primarily made up of phospholipids as well as cholesterol. And then there are some proteins that are inserted within the cell membrane, right? And these proteins um, have various functions. They provide attachment sites for the cytoskeleton. They play a role in cell signaling. And one of the functions that we talked about towards the end is that they play a role in transporting material across the cell membrane. And that is the main concept that we're going to be going through today, like how to move things across the cell membrane. As you will see, a lot of these processes does not involve energy. But in some cases, if the molecule is too big, or if you're trying to push it against its natural course of movement, then you would need to supply a bit of energy. And so we will spend a few slides explaining where cellular energy comes from and the molecules that are involved in capturing some of these energy. All the chemical reactions that occurs within a cell are collectively known as the metabolism of the body. And they are separated into two kinds of reactions. The reactions that involves in breaking things down, as well as the reactions that are involved in building things. When we try to break things down, we call that catabolism. Okay, sometimes we say they are part of the catabolic pathway, the catabolic reactions. Cata is the same root word as catastrophe. In catastrophe, everything breaks down, right? So in catabolism, you are breaking things down as well. And in doing so, when you're breaking these things down, in breaking these things down, you are harnessing the energy from these molecules that you're breaking down. And so energies are going to be released. On the other hand, when you are building molecules, that's called anabolism, right? Or we say it belongs to anabolic pathway. Building things takes energy. Okay, it requires effort. It requires time and investment to build things. Are you guys familiar with? Hold on, I'm just trying to copy a picture over to show you, or I'll just pull it over, right? Like you guys know this game, right? This is called like Jenga, right? Like if you want to build a tower, it takes time, it takes effort, it takes energy. To knock it over is easy. You just pull the wrong block, and then the whole thing just comes tumbling down, right? And then it creates sound, and it falls down, it hits the table. All the energy is released, right? So catabolism is like bricking the tower. Not much effort is required. It releases energy in the process. And anabolism is like building it back up. Okay. So within the body, when we eat food, what is exactly in the food that we eat? Anybody want to tell me what are some of the molecules that are found in our food? You should know. We spent a whole lecture talking about it.
Say catabolism.
Sorry, what is it in it again? Okay, I see someone said protein. Okay, what else is in the food that we eat? Yeah, there is lipids, right? Lipids. Basically the macromolecules that we talk about, right? Okay, and then if the thing that you're eating is alive, was alive, then it has a nucleic acid, right? So all these macromolecules needs to be broken down. They need to be metabolized, and that's where the catabolic reactions are going to be useful. In breaking down the food, it releases energy, and these energies will be captured and stored as ATP. That's the usable form of energy in the cell. Now, we are not very good at capturing all those energy. In fact, we are not efficient. A lot of the energy is lost as heat. Okay, so whatever we don't capture will be used to warm our body. Okay, so you feel warm, you feel warmer, especially on like a cold day. You have a nice meal afterwards. You feel much better, much warmer, and that is primarily due to the heat that's lost as we are breaking down the food. So you break down the food and we get all these building blocks. Okay, so when you break down the proteins, maybe you will get amino acid. When you break down the carbohydrates, you're going to get glucose, right? And all these building blocks can be used to make things that are actually needed in the cell, right? So the ribosome works with the rough ER to make proteins. Where is it going to get the amino acid? Well, it's basically going to get it from the food that you have broken down, right? And then as you're building it, you would use the energy that was released and captured, and now you use it in the anabolic pathway. So basically, the catabolic pathway provides energy and resources to enable anabolic reactions. Okay? Not only does it provide the energy to do so, it also provides the raw material, right? The building blocks that are necessary to build whatever you need. Any question about this concept of catabolism and anabolism, how they're related, for example? One of the major catabolic reactions in the body is cellular respiration. That is where you take the glucose and you react it with oxygen to produce carbon dioxide, water, and you will capture the energy in the form of ATP. Does anybody remember which organelle is responsible for producing energy in a cell? What did we learn last class? Which organelle is responsible for producing energy? Oh, Justine said mitochondria. That's correct, right? So that's where the process occurs, right? Here you can see a picture of the mitochondria. That's where you will be making the ATP molecule. So here's the structure of ATP. It stands for adenosine. The adenosine is the same as the adenine in DNA. Okay, remember DNA has A, C, T, and G. The A is the adenine. So we see the double ring structure right here. It's connected to a ribose sugar. And then it's connected to 3-phosphate. Tri, right? Tri means 3. And this last bond that's connecting the phosphate together, the last 2 phosphate together, this is a very energetic bond, okay? High energy bond. When you break that high energy bond, energy will be released, and you can use that energy to do various things. Okay, so here is a equation that shows you what's going on. We have the ATP triphosphate when you add a water molecule, you chop off the last phosphate, and then you release the energy. You can use this energy for anabolism, for example, right? For building proteins, for building glycogen, for building whatever you need to build. Okay. Without the P, now it only has 2 phosphate, so we call it adenosine diphosphate, ADP. Essentially, you would have a pool of ATP in your cells, and then you can use them up, right, by ripping off the phosphate, and then now is empty, is not charged. It doesn't have energy anymore. So how do you recharge it? Well, you can take the energy from catabolic reactions. Again, these are reactions where you would be breaking your food down, and in the process, you release energy. So those energy can be used to recharge the ADP so that you can create ATP once again. And then you can use it up, and then you can charge it, use it, charge it. It's like your phone. When it's fully charged, it's ATP. And once you drain it, it's no good. You cannot use it until you recharge it, right? So we recharge the phone by plugging it into the wall. But in the body, to recharge the ADP, we need to eat. Okay? By eating, we provide our body with the food molecules, the lipids, the proteins, the nucleic acid. And when we break it down in catabolism, we can harness the energy from them, and we use that energy to recharge the ADP. Okay? What else do we use ATP for other than building things? Well, we can use it for mechanical work. Mechanical work means to move something. Okay, involves movement. If we're looking at a single cell, maybe it has flagella and it needs to, whip them to swim around. That's movement. That requires ATP. As a whole organism, to move our arms, right, for the hearts to beat, to breathe, right? All these things involve movements, and all these things involves the use of ATP. Without ATP, the muscle would not be able to relax. It would contract and it would get stiffened, and you can no longer move, right? When a person dies, they no longer breathe, obviously, and without oxygen, you cannot make ATP, and that's why the body will stiffen up when a person dies, right? That's called rigor mortis. Some of you might have heard of that before. We can also use the ATP for transport work, right? To move things against their concentration gradient. I'll talk more about what that means later. And here we have chemical work, okay? We previously talked about enzymes. They are able to speed up chemical reactions by lowering the activation energy, right? But nothing is free. Something has to be supplying that energy. And in some cases, it would come in the form of ATP. Excuse me. Remember, I used the analogy of like a scholarship, right? To lower the tuition, the enzyme is like the scholarship, right? But then like, that's not free money. Someone has to be paying the money. Someone has to be donating that money, right? So in the same sense, that missing chemical energy, something has to be paying for it. And in a lot of cases, that something is the ATP. Okay. So let's take a look at this question. Read it first, and then I will launch the poll in a second. Really? Only 3 people wants to answer? Okay, that's fine. So here, catabolic reactions, they consume more energy than they produce. That's not true, right? We need to extract the energy from them, right? And so they produce more energy than they consume, actually. Breaks down polymers into monomers. That's true. We know that. Produce excess heat to warm the body. It was on the diagram. And it's not restricted to human cells. Okay, plant cells, fungus, they are able to perform catabolic reactions as well. All right, we're gonna move on to the type of transport now. First, we will talk about diffusion. It's the simplest kind of movement of substances. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. So in case you have trouble understanding what high and low concentration means, we have 2 containers here. Let's fill them up with the same amount of water. Let's say I add a lot of salt here, right, and then I add a little bit of salt here. So this one would have high concentration. We use square bracket to represent concentration, okay? And over here, because it doesn't have a lot of salt, this has low concentration. Okay? So this would taste saltier compared to this one here. Now, if I connect the tube, if I connect the tube with the tube, then naturally the salt is going to move from high concentration to low concentration. You don't have to do anything to encourage this or to promote it. It will just happen on its own, spontaneously. All right. A way to visualize this is, for example, we add a few drops of dye into the beaker, and the dye is very concentrated, and then there are no dyes elsewhere in the beaker, and so the dye will naturally spread out, right, from area of high concentration to areas of low concentration. And if you just let it sit there for some time, eventually it will fill up the entire flask, right? So that is what we call diffusion. You can also imagine, taking air freshener, the Febreze or something. You spray it, in one corner of the house. Soon you'll be able to smell it in the other end, right? Diffusion, from high concentration to low concentration. And the molecules will move down their concentration gradient. A gradient is basically a difference between 2 areas. Okay, so if you have 2 areas with different concentration, the molecules will spontaneously, on their own, go from high concentration to low concentration. That's what diffusion means. Okay. Here are some factors that affects the rate of diffusion. Rate of diffusion is how quickly the spread occurs, right? So first factor that affects it is size. If it's really small, like this O2 molecule, it's able to move quickly and therefore able to diffuse faster. If the molecule is more bulky, like this one, this is glucose, right? It moves slower, and the rate of diffusion is going to be taking longer. Second factor that affects rate of diffusion is temperature. Higher temperature will allow molecules to move faster. They are more energetic when there's higher temperature, and then they spread a little bit faster. Imagine walking on the street on a hot summer day, and it's like garbage day or something, right? You can smell the garbage, right? It's very unpleasant. But you know, the hot temperature causes the scent to, you know, go into the atmosphere and it spread quite quickly. Not so much on a cold winter day, right? You don't really smell much because the cold temperature prevents it from diffusing as quickly. All right, so two factors, size and temperature. The third factor is the steepness of the concentration gradient. So here we look at two areas. The area on the left has a lot of these dissolved particles, right? So we can call that, like say 80% concentrated. And there's nothing here, so that's 0%, right? So the difference here is 80, right? So the molecules will come over very quickly, right? Not all of it, right? They will just come over until both sides are the same, right? Over here, right, we have less particles here. So let's say that's 60%, and this is like 40%, or like, you know, 30%, something like that. The difference is only 30%, right? Compared to 80%. So they will still go, but it will take a longer period of time for them to become the same. Okay. So depending on how steep, how great the difference is between the two areas, that will affect the diffusion rate. Okay. The greater the difference, the faster, the higher the rate of diffusion. Think of this as a really steep ramp. Okay. You put a ball here, right, and let it come down. And over here, it's like a shorter ramp. Okay. Like not as high. Which ball is going to come down faster? Hopefully, common sense tells you this is going to be much faster than that, right? And that's because this is steeper. The ramp is steeper. The difference in height is greater compared to here. So same thing when it comes to concentration. All right. The last factor is charge. All right, this one, maybe I need to draw it out for you. You don't have to draw this. We're gonna have a lot of drawing coming up in a bit. But let's say we have two kinds of particles here. Okay, let's say we have these ones here. Okay, and then down here we have the same particle, but now I'm gonna give them a charge. Okay, I don't know why they don't show up. Let me just try it one more time. All right, so let's say here is one chamber, and then here's the other one, right? Now, based on what I told you, both of them is gonna move down, right? Both of them are gonna move to the right, from high concentration to low concentration, right? But which one will move faster? The top or the bottom? What do you think? Which one will have a higher rate of diffusion? AppNet's at the top. That's right. That's one answer. Anyone else? What do you guys think? Top or bottom? Which one is faster? Okay. Well, like charges repel each other, right? Positives don't like to be next to other positives, right? They repel each other. So over here, because there's so much positive here, they will be pushing away from each other. And so that repulsion force will actually cause them to move away from each other faster. Okay, so this is going to be faster compared to the top. Okay, same thing. If they're all negatives, then it will push them faster as well. All right. Hopefully that makes sense. And if not, feel free to ask questions. Okay, I think that is it. Now we're gonna spend quite a bit of time taking notes. All right, so go get some paper And we have a lot of things to write. Sorry, having a bit of a cold here. So first one we're gonna do is osmosis. Okay. Osmosis. This is the diffusion of water. Diffusion of water. So let's take a look at this. Let's say I have a big container here, and I'm gonna put in a barrier. Okay, more on that later. And then on one side, I'm gonna dissolve a lot of solute. And then here I'm just gonna have like maybe a few. And then of course I'm gonna fill it up with water. Okay. So all this is water, huh? You don't have to color it in. So which side has higher concentration? What do you think, left or right? Which side has higher concentration based on my drawing? Yeah, the left side, right? So high concentration, low concentration. Now, if I remove the barrier, okay, if I remove the barrier, you don't have to erase yours, okay? If I remove the barrier, which way will the red circles go?
Right.
I'm sorry, what is it?
To the right.
To the right. Yeah, there's no trick question, right? This just goes to the right, right? That's called diffusion, right? Excellent. Now I'm gonna put the barriers back in. Now this is a special barrier, okay? It has some holes, but the holes are really small. It will only allow H2O to pass through, okay? So even though the red circles want to go to the right, down their concentration gradient, they cannot do it because the barrier is blocking them. You see the hole is really small. The red circle will not fit, right? So the only thing that can move in this case is the water. Which way will water move? Okay.
Right.
Let's do a poll here. If you think it's gonna go to the right, choose A. If you think it's gonna go to the left, choose B. If you think no movement, you can choose C. Let me repeat the question. The red circles cannot move because the barrier is blocking it. Only water can move. Which way is the water going to go? That's my question. Okay, please participate. Anyone else? Okay. Well, unfortunately, most of you got it wrong, but that's okay. That's why we are in class, right? So over here, we can see that there is a higher concentration for the red circles on the left and on the right. And you guys correctly said that the red circle is going to go and move to the right, right? If I remove the barrier. But when the barrier is in place, it's impossible for the red circle to move, right? So the only thing that can move is the water. And water will move actually to the left side to try to balance out the concentration. Think about it. When water moves to the left side, you will be diluting the solution, right? Okay. Like if you have a glass of juice or something, orange juice, and it's too sweet, you can add water to dilute it, right? So as the water shifts to the left, it will dilute the left side. And when you are removing water from the right side, you will be concentrating it, right? I don't know if you guys cook, but like if you have a sauce for your food, you need to heat it up to evaporate the water, right? To reduce the sauce so it gets more concentrated, right? And that's what's happening here. As the water goes from the right side to the left side, you will be diluting the left, in other words, bring the concentration down, and then you'll be concentrating the right side, which means you bring the concentration up until the two sides are the same. So water will move to the left. Okay, in fact, water will always move towards the more concentrated side. You can try this if you want, okay? Put a spoon of salt in your mouth and hold it. Like, don't swallow it, okay? It's gonna not be good for you if you swallow it. Just put it in your mouth. Hold it there for like 30 seconds, okay? You will feel the saliva being drawn out by the salt. Okay? You'll be like drooling all over, right? That's an illustration of this effect, right? Higher concentration attracts water towards it. Any question about this? Anything at all?
Sir,
Yeah?
sir,
Yeah?
can you please explain osmosis again? You just said the diffusion of,
The diffusion of
is
water.
it,
So
is it basically osmosis is water? I don't know.
osmosis is basically looking at how water moves when you have two different areas with two different concentrations.
Okay.
Okay. Right, you understand? Like it will suck it towards one end. And it basically will move towards the more concentrated end all the time.
Okay.
Right? It will draw the moisture towards it basically. So here is another view. We have a U-shaped tube here, and then there is a membrane at the bottom, right? That separates the two solutions. It's more concentrated on the right side here, high concentration. This is low concentration. And over here, as I told you, the water will be drawn to the right side, right? Make sense? It will be drawn to the right side. As it goes to the right, the water level will go up on the right side, and then the water level will come down on the left side. And this will continue to happen until the concentrations are the same on both sides, right? As water moves to the right, the left side gets more concentrated. And the right side gets diluted. Okay. So the water level will actually not be the same afterwards. Right? Please pay attention to this concept. You will see a question like this on the test. Any other
Sir,
Yep,
I have
go
a
ahead.
question.
Mhm,
I was sorry, because you said test, I was looking at the course outline,
mhm.
but in the beginning, I remember you saying that we would have like the quiz and then we forget about it. But on the outline, we have like
No,
a
no.
we have
When
two tests.
after the test, you can forget about it. Yeah.
Oh,
Yeah.
okay, okay.
So quiz 1 is 1 and 2, quiz 2 is 2 and 3, test 1 is 1 to 5. Okay, and
Okay.
then afterwards you will not be tested on 1 to 5 again. Okay.
Oh,
Yeah. All right. Before refrigeration was invented, people preserve food by salting them, right? Okay, salting them means you coat the food in salt, and the salt removes the moisture, removes the water from the food, right? And without moisture, without water, there's no bacteria, no microorganism to spoil the food, right? That's why you can keep them for a long time. All right. Now we're gonna take another note. Now, if you have a new page, or even if you're using the same page, I want you to take the note in 3 columns. I'm gonna show you 3 cases, okay? Case 1, case 2, case 3. It's better if you draw them side by side so you can compare what's going on. Okay, so the title is tonicity. Okay. Movement of water when a cell is placed in different environment. Okay. And I'll show you 3 different scenarios. First case. First case is called isotonic. Isotonic. Okay. So in this case, we are going to have a cell, and we're going to put it in a beaker. And the concentration on the inside is same as the outside. Okay, so concentration outside equals to concentration of the cytoplasm. So which way will water move? What do you guys think? Anybody? No movement? Okay, very good. So because the concentration is the same, right? The amount of water that's moving out is same as the amount of water that's moving in. It's not that there's no movement, but they cancel each other. Okay, so rate in equals to rate out. Okay, no net water movement. Okay, no change in cell volume. So it's hard to visualize, right? You can think of it like this. Let's have a basin. And I am going to turn on the tap. Okay, so the water's coming in. It's gonna fill up the basin. All right. But I'm gonna poke a hole here at the bottom of the basin. So water's gonna leak out, right? So in this case, water is leaving at the bottom, but at the same time, water is coming in. But the rate at which the water is coming in is the same as the rate it's leaking. So what's gonna happen to our water level? What's gonna happen to the water level? Is it gonna go up, go down, no change?
No change.
No change, that's right. So in the
Well,
same,
sir,
Mhm.
sorry, doesn't it depends on the size of the hole you're putting in there?
That is a great point, right? So in this particular case, I make the hole just the right size so that it's gonna be leaking out the same way. Okay, so if it's coming out the same rate as this, then it will be no change. Later on, I'm gonna make the hole bigger, as you will see.
Okay.
All right. So over here, no change in water level. And in the same sense over here, because the amount of water that's coming in is same as the amount that's leaving, no change in cell volume. For animal cells, including us, we are animals, right? They are happy. We love this. Isotonic is the best. Okay. For plant cells, they are not the best in isotonic. They're okay. They're not like upset or anything. They'll survive, but not the best. Okay, this is like if you forgot to water your plants for a couple of days, right? They wither a little bit, right? The leaves kind of crunch up, wrinkle up together. Not the best, but they'll live. Okay. Any question about isotonic? Gonna give you two other examples, which are more confusing. So I just want to make sure that you are all okay with the isotonic. Great. So again, if you're doing this on your paper, right, you have your isotonic here, you can write the next one next to it so you can compare, right? I cannot do it because I'm going to run out of space, right? So I'm going to do it right here. Next one is hypotonic. I want to put a big O here. Okay, hypotonic. Sorry. So hypotonic goes like this. Let's have a beaker. And I'm going to put our cell in it. In this case, you have a lot more concentration on the inside than outside. Okay. So concentration, square bracket means concentration. Outside is less than concentration of the cytoplasm. Cytoplasm is the inside of the cell. Right? This symbol means less than, right? So in this case, which way will water move? Into the cell or out of the cell?
Out the cell?
Out of the cell? Okay, anyone else? Did you agree? My treat said into the cell. Okay, let's go back here and see what it says here, right? Water will always move towards the more concentrated side.
Into the cell.
Into the cell, that's
Yeah.
right. So over here, you will have, you will still have water leaving. Okay, some water will leave, but the point is there will be more coming in. Okay. So rate in is greater than rate out. So the net water movement into the cell. All right. Let's draw our basin again with the hole in the bottom. This time, we're going to turn on the tap all the way. Right? So it's really, really coming down. But it's the same hole as before. Right? So it's still leaking the same. So what's going to happen to the water level here? What does common sense tell you? If you turn on the tap all the way, even it's leaking, it's going to increase. That's right. Okay, going up, going up, going up like that. And eventually what's going to happen? It's going to overflow, right? And so that's what happens to the cell, right? Cell volume increases. Okay. If it's an animal cell, this is bad news. Okay? They're going to swell up so big and then they're going to explode. Right? They're going to burst. Right? We call that they lyse or they experience lysis. Okay, Lysis, lysis. Okay, so it will die, the animal cell. Plant cell. Do you expect the plant cell to burst as well? Or not?
No,
Why
No.
not?
but it's going to overflow.
The plant cell is going to overflow? Okay. What blessing? Why is the plant cell not going to burst? You said no. Oh, Justine answered already. They have a cell wall. That's right. Okay, so they're happy. Right. So this is the plant cell. All the water will fill up their vacuole, and then it will be pushing against. It'll be pushing against the cell wall. Okay. So cell wall prevents bursting. Okay. The plant cell are the happiest in a hypotonic environment. Okay? The water will make the plant stand up, all the leaves will open, they will be able to capture as much sunlight as they want. Okay? So this is hypotonic. Remember the big O here, right? It shows you that the cell is swelling up. All right. Now, some of you might know like the video game console, right? This was like, I don't know, almost 20 years ago, I think 15, 20 years ago. The Nintendo Wii, right? Okay, so when it came out, it was everybody wanted one. And then there was this radio station. It hosted a competition called Hold Your Wii for a Wii. That's what the name of the contest is. So like the contestants, they had to drink as much water as they could and hold it, not go to the bathroom. And if you're the last person standing, you will win the console. Okay, and so one of the contestants, a lady, she won. Okay? She was able to drink a lot of water. I don't know how much, but like quite a bit. And she was able to hold it, and then she won the console. But then she was found dead hours later in her apartment. So how does drinking too much water in a short period of time cause someone to die? Based on what I explained to you, why do you think that is the case?
Because You told us animal cells cannot really retain water, like it has limit, and if it gets to a point, it bursts.
Exactly, right? So like when you drink too much water, you're diluting your blood. It makes your blood hypotonic to the cells, right? So you have a lot of red cells in your blood, right? Red cells are important for transporting oxygen, right? And so if they started bursting and dying, you have no cells left to transport oxygen. You die, right? Having a lot of water in your blood also dilutes your electrolytes, your sodium, your chloride, right? All that stuff is important for normal bodily function. They are responsible for allowing neurons to communicate with each other, right? If the concentration gets too low, your brain can't communicate to your heart forgets to beat and you could die, right? So that's water toxicity, okay? If not treated immediately, it could be lethal. Are we okay? Isotonic, it's the same. Hypotonic is too diluted outside. All the water is coming in. Cell might burst. Okay. Any questions? Please ask.
Excuse me, sir, for our test, like is it that you're going to draw
No drawing for the
the
test. Yeah.
diagram
There's no
or
drawing. Yeah, no drawing on the test. Okay. The quiz, the test will be exactly like the first one. Right? You will have the multiple choice question. You will have multi-select question. You will have some short answer questions. In fact, I already told you the short answer question for quiz number 2, right? Okay, I told you that last class.
Yeah, thank you, sir.
Yeah. Okay, well, let's take a look at the next part here. We will have our third case. Our third case here is hypertonic. Okay, hypertonic. You can probably guess what it's like and probably can finish the note yourself, but here we go. In hypertonic situation, you have more solute outside than inside, right? I hope this conveys the situation to you. Right, so concentration outside as in the surrounding environment, right, is greater than the concentration of the cytoplasm. So I want to ask you again, and hopefully you will get the right answer this time, all of you. Okay. Is the water going to come in or is the water going to go out?
Gonna go out.
It's going to go out. Okay. Anyone else? Does everybody agree with Patrice?
Out.
Out. Excellent. Okay. Because water moved towards the more concentrated side, right? Okay. I'm happy that you guys will get it. Okay. I assume the rest of you also get it. Okay. Like I assume you're yelling the answer to the screen. Out, out. All right. So we will have water leaving, but remember, it doesn't mean water is not coming in. They are. It's just that you have more leaving than they're coming in, right? Okay. So rate in is less than rate out. Okay. So let's draw our basin again. This time we have a gigantic hole at the bottom. All right. And we're just going to leave our tab as normal. So water is coming in. But boy, we have a major leak here. Okay. It's just coming out like that all over the place. All right. So leaking. And then this is coming in. And again, what does common sense tell you? Is the water level going to go up, go down, no change? Well, hopefully you know it is going to be going down, right? So here we have net water movement. Net means like in total, right? like your net income, right? It's like you take out the money you make, subtract the money that you spend, right? So here, net water movement, you subtract the two, is going to be out of the cell. All right. And cell volume decreases. Cell volume decreases. For animal cells, it's okay. Not the best, but at least we're not like bursting and dying. So, we are okay. The cell is gonna look like this. It's gonna shrivel up like a raisin. Okay, all the water will leave, and this is called plasmolysis. Okay, plasmolysis shrinks. Plant cells, they don't like it. Okay, you forgot to water your plants for like more than a week now. It's really shriveled up. Okay, even the stem is like starting to bend down. If you don't water it soon, it's not coming back. It's gonna die. So 3 scenarios. So talk to me. Can you give me an example? So like the previous one, right? It was like drinking way too much water, right? What about here? What might be a possible cause of creating a hypertonic environment for ourselves? Something that could happen. What do you think?
Like, is it like what will happen to humans or in this scenario?
What happened to what? I'm sorry.
Like, please,
Repeat the question.
can you please reconstruct the question because it's
Sure.
not-
I'm just asking, right? Like what everyday scenario is going to cause this kind of situation to occur in the body, right? Where your cell all shrivel up. That's right. No need to think too hard, right? Abnet said dehydration, right? You don't drink enough water. Most of us don't drink enough water. Okay. Like, especially if you're running around, to class, you're doing errands, right? Like you might drink a little bit in the morning and then you forgot to drink until lunch. So like our body gets dehydrated, especially if like really hot outside, you sweat a lot, you become dehydrated, and then the cell shrink up, right? Okay. So we have special cells in the brain that will shrink when we are dehydrated, and that shrinking sends a signal throughout the brain that tells you, you're thirsty, you should go drink something. Body is really dehydrated. Okay. So another story. Do you guys know Red Bull? Red Bull.
Yes.
Yeah, Red Bull is a caffeinated drink, right? Okay, there are other ones, the ones with like the 3 green claws. Forgot what that was called. But like these caffeinated drinks, when it was first came out, it was a popular drink in nightclubs. Okay? And this is based on like news article that I read. People like to mix it with alcohols and then drink. I guess like the caffeine would keep you awake, right? And you can party all night, right? And drink alcohol at the nightclub. But what happens is the caffeine is a very potent um, diuretics. It's something that makes you pee a lot, right? Like you would know if you drink coffee. Drink a cup of coffee, within an hour you're gonna pee it out, right? So when you drink these Red Bull, the caffeine causes you to go pee, and if you drink a lot plus the alcohol together, it causes you to become dehydrated. So these people who are adding the caffeine, adding the Red Bull to the alcohol, they get thirsty real quickly. Okay, so when you get thirsty, you should be drinking water to rehydrate yourself, right? But what happens was people continue to drink more. They're like, oh, I'm so thirsty, let's have another Red Bull, right? Okay, so they do that. And after the second or third one, it turns out your body starts to get really dehydrated, despite the fact that you're drinking something. But the fluid that you're drinking is not replenishing your water. It's causing you to, actually lose more water as a whole. And people actually die from severe dehydration as a result of that. Caffeine is also dangerous for another reason. If you consume too much, it could actually cause arrhythmia. Okay, arrhythmia is irregular heartbeat, and your heart basically goes into this spasm, and you could basically drop dead without warning from consuming too much caffeine. That is. Okay. So are we all together? Isotonic, hypotonic, hypertonic. Tell me anything you want me to do and say to make you feel better? I feel like
Sir,
I've confused you guys and stressing you guys out. Yeah, go ahead, Patricia.
I have a question.
Mm-hmm.
I don't understand why you're drawing the sinks with the hole at the bottom, right?
Oh, the sink is just for you to visualize what happens. Like, so when you
Is it that the cell membrane, does that represent the cell membrane? Is it getting bigger or?
the hole at the bottom represent water leaving the cell, right? And then the tab at the top here represent water coming into the cell. In the case of a hypertonic situation, more water is leaving the cell than they're coming in, right? And if this is happening, overall your water level is going to go down, right? The water level is analogous to the cell volume. Because you're leaking more than you're replacing, overall the cell is going to shrink. That's what the diagram represents.
Yeah, but that's due to the concentration that's outside.
Exactly, right? Because there's more stuff outside. That's why you are drawing more water out of the cell, right? Causing it to shrink. Okay. And I was saying, right, like, if you think of this as a red cell, the blood is basically the environment, right? If someone is very dehydrated, not drinking enough water, that's what will cause the blood concentration to increase drastically. Okay, does that answer your question?
Yeah, but I still kind of think your sink diagram is
Confusing?
wrong.
Is wrong? It's like, well, I can tell you it's
I
not wrong, but go ahead, go ahead.
Wrong, but I just don't understand why you're making why the hole can stay the same size, why you're making it bigger compared to the other.
Okay, so like over here, right?
diagrams.
Over here, I'm turning on the tap all the way, right? When I turn on the tap all the way, do you agree more water is going into the basin than it's leaving?
Yeah, I do.
Yeah, and then the water goes up, right? So, the water
compare your tap to the cell membrane.
The tap is not the cell membrane, okay?
Oh.
There's no cell membrane here. Okay, well, if you, I guess this here is the cell. The basin is the cell, okay? And the water in the basin is the size of the cell. So the size of the cell will either get bigger or smaller depending on how fast water is coming in and how fast water is leaving. Is that point okay?
Yes.
So in an ideal world, the amount of water coming in is same as the amount of water leaving, so no change in our cell, right? That's the isotonic environment. But when the environment changes, when it's hypotonic, then it turns out water is coming in faster than they are leaving. That's why the cell is getting bigger and bigger and bigger.
Okay, now I understand the diagram.
Okay.
I got it. Thank you.
Thank you for bringing that up. I'm sure there are other people who are, confused. Hopefully that clears it up. All right, we're gonna take a break. We'll continue at 1:41. Don't leave, okay? I'm gonna talk about the lab right after the break. All right, like, I mean, you can go for your break, but like, don't sign off, okay? Come back. I'll tell you about the lab. All right, see you in a bit, guys. Hello, I'm back. Let's take a moment and talk about the lab. So, let me switch to student view here so you can see exactly what you will see when you log in. I'll post an announcement after class to direct you to things. But basically, if you go to content and virtual labs, content, virtual labs, you will see the two labs here, the beetroot experiment and the microscopy. So we will be doing the beetroot experiment. There is a video that you have to watch. It's a short video. It's only 8 minutes long, and it's someone doing the lab. Okay, I've been trying to film our own lab, like me and other profs doing the lab and then doing the narration, but that is taking some time. We got the footage finally, but we still have to, edit it, put it together, and stuff like that, and that is taking longer than expected. So for now, you just have to watch someone else do the lab. And like I said, it's a short video. It's only 8 minutes. You should watch it before you fill in the lab report, okay? Because the lab report will be based on the video. A lot of the questions that are being asked are based on the videos content. All right. You won't know the answer. You can't just search them up online. Okay? If you search it up online, the answer that you find will not be specific to the video, and if you just copy it, it'll be wrong, right? So, here, I mean, you can find the video on eCentennial, right? Content, virtual lab, and then lab 1. But I'll post it in the chat box if you want to, bookmark that yourself. Now, once you've watched the video, you will be doing the Beetroot lab. Okay, so click on the Beetroot lab, and it's a Word document. All right, now you cannot type in anything right now. That's because you have to download it first. Okay, you have to download it. So download a copy onto your computer, and once you have it, you can open it up. All right, just give me a second. I'm trying to open it right now. So there it is. Okay, so you will type in the answers of the question. Right, first question: what is the purpose of this experiment? Right, so you can just type in the answer directly. The purpose of the experiment is to examine the effects of temperature on membrane permeability of beetroot cell. That's it. You type that, I give you 2 marks. Okay. In fact, why don't you just type exactly what I have here? All right. You don't have to make up your own thing. Just copy this. I give you permission to copy it. There, 2 marks. You're welcome. Then what is the hypothesis? Okay. Remember, when you write hypothesis, it should be a statement. It should be an answer, right? So if the purpose is to examine the effects of temperature on membrane permeability, right? What would be a good hypothesis? Someone give me a good hypothesis, please. You can type it out. You can unmute. You don't have to copy that right now, okay? Like, I mean, like it's gonna be in the recording, right? You can go back to it and watch it later. But give me a hypothesis, please. Nobody knows an hypothesis? Okay, well, that's all right. I'll let you think about it. So this is what you have to do between now and Tuesday. Okay, you're gonna go watch that video and you're gonna go answer as many of these questions as possible. Okay, and then Tuesday, we're not gonna be able to finish the class today. Okay, there's too many slides. We'll finish up the slides on Tuesday, and then I will help you with some of the harder questions. Okay, you can come prepared though. You have to come prepared though, right? By the time you come, you have to have watched the video and you have to have attempted some of these questions. I mean, don't show me your answers, but then like you would know which one are the harder ones, right? And where you're getting stuck, right? So I'll be helping you with it. There is a question where you need to plot things. Okay, I was told you guys already learned how to graph in your computer class. Is that correct?
No,
You didn't learn how to graph. You didn't learn how to use Excel to graph in computer course.
well, a pie chart.
Yeah, pie chart.
You did a pie chart, but you didn't do like a line graph?
I think there's a video of it.
Okay. Who's your computer teacher?
Mr. Louis.
Oh, okay. Louis,
Lou.
yeah. Okay.
Mm.
All right. So that's fine. I mean, like if you know how to do it, great. If you don't know how to do it, there is a link that shows you how to do it. If you still don't know how to do it, that's okay as well. Come to class on Tuesday. I'll show you how to do it. All right. So you can do it on Excel if you have Excel on your computer. If you don't have Excel, you can use Google Sheets. Okay, Google Sheets are free for everybody anyway, so that's not a problem. But I'll show you, okay, on Tuesday's class. So is that clear? What needs to be done? This is 10% of your final grade. You do well, it will boost your grade. Okay, and people usually do well. The only ones who don't do well is they didn't watch the video and then they try to copy answers from internet that makes no sense. And you know, don't leave it until last minute, right? So no class next Thursday. You do not come to class on Thursday. You just use the class time to finish up the report and you hand it in on Thursday. Okay, there will be a Dropbox on Thursday on eCentennial. Like if you go to assessment, I'll post a link, okay? But like if you go to assessment and assignment, you will see the two folders. The folders are not open now. I'll open them later, okay? And you just submit it electronically, okay? When you submit it, please submit the file as document file, .doc, .docx, or PDF. Does any of you use a Mac computer here? Any Mac users, Apple
Yes.
users? Yeah,
Yes.
if you use Apple's, you must convert it into PDF before you submit it. Okay? If you don't know how to do it, find out online, find out from YouTube. They will teach you how to do it. If you submit it using your Apple format, the page format, I cannot open it. Okay? The college did not give me an iPad. So I cannot open it, I cannot grade it. All right. And then by the time I email you and say, yeah, I cannot open it, and then maybe you don't check your email until a couple days later, it's already like 5-6 days later. Okay. Like you will have a 0 grade by then, right? So make sure you submit the right format. Okay. The PDF, please. What if you have, yeah, that's fine on your Mac, right? So make sure you save it. Make sure when you hand it in, the file extension is .doc, right? Okay, or .docx. As long as it's not .page, then it's okay. All right. If anything, I will email you and when I can open it, but it's just like, you got to check your email quickly, right? Okay. Otherwise, it's like you wouldn't know. And then you get a 0, and you're like, why did I get a 0, right? Anyone else have any questions about what you need to do for the lab and what's going on next week?
I just want to be clear.
Sure.
We're doing beetroot experiments.
That's right. But
And
not,
it's
you're
due-
just watching someone else do it, right? You're
Yeah.
not doing it yourself, right?
Yes. And then it's due on Thursday next week.
That's right. So you just download this document, you answer all the questions. The questions are based on the video, right? Okay, I just answered one of the questions for you, right? Okay, you can just copy this, no problem. All right, so the Tuesday class, we will do more, we'll go through the lab more, but by then I expect you to have done your work already, right? And then no class next Thursday. You don't have to come. You just finish up your lab and hand it in. If you finish it before then, great, you get a day off from me
But what time the lab is due on Thursday?
before midnight, okay?
Mm.
Yeah. Are we all good?
So do we have quiz or test next week?
No, next week is just look at the calendar, right? Next week is just the beetroot, okay? And then the following Tuesdays will be quiz number 2, which is lecture 3 and lecture 4. Okay. And then before you know it, we will have the test, and then we'll be done half the course. Okay. Very straightforward, the assignment. You just have to put in the time to do it. Okay. And don't copy each other. Okay, that's very important, right? Use your own words. I know you guys probably have your own chat group or something online, right, with other students. Don't copy each other. Okay. I have very good memory. If I see an answer once and then I see it again, couple assignments later, I will remember it. Okay, and then I will have to talk to you guys, right? I don't want to do that. Just use your own words. Okay. All right. So that's that. Let's go back to our lecture. So this is the red blood cell in the body, and it shows you what happens when you put the red cells in different environment. Right? So isotonic is the normal environment. They look just fine. If the person is overly hydrated, too much water, then it's hypotonic. The cell swells up and it might burst. And then hypertonic is when the person is dehydrated. You can see all the red cells are shriveled up. Okay. So why do we have to know this, right? Well, I assume a lot of you are going into a healthcare related field, and you know, in your work, you might have to apply IV to your patient, intravenous solution, right? So for healthy patient, the IV fluid is typically isotonic to the cell, which is what you want. Like, you don't want the cell to swell up, you don't want them to shrivel up, you just want to rehydrate the person without affecting the cell too much, right? So typical saline is 0.9%. Okay, 0.9% means 0.9 grams of salt per 100 milliliters of water. Okay, that's the isotonic IV fluid. And it's mostly for hydration purposes. So people who are bleeding too much, you want to rehydrate them. People who are vomiting excessively, or if they have diarrhea excessively, right? You want to give them 0.9% for hydration purpose. Right? Some people are severely food poisoned. They can't keep anything down, so they cannot drink the fluid. Then you have to give them an IV line to maintain the hydration. Sometimes you might want to give them saline that is hypotonic. Okay, hypotonic, 0.45%. And in this case, the solution is more diluted than your cell. It's going to create a hypotonic environment. And that's useful for treating things like hypernatremia. Hyper means too much. Natri is sodium. So too much sodium in the blood. Emia is in the blood. Okay, so too much sodium in the blood. Or in some cases, when people have diabetes, the body thinks they're starving and they start to break down fat as a way to harness energy. And a byproduct of breaking down fat is it creates a lot of ketones. Okay, and ketones are acidic. So ketoacidosis means your blood is getting acidic. And so by diluting the blood, you are lowering the concentration of these ketones, okay, making your blood normal pH again. If someone has hyponatremia, not enough sodium in the blood, right, like that person who drinks too much water in a short period of time, right, if they rush her to the hospital quick enough, right, they could infuse her with 3% hypertonic saline, right, and that would help undilute, if you would, her blood, right, by introducing more salt into it. Okay, so different scenarios require different kinds of saline solution. That's the point. All right, we're gonna switch gear and talk about transport, and there are two main categories of transport. There is what we call active transport, and then there is passive transport. Passive transport does not require energy from the cell. Okay, it just kind of happens on its own. It happens spontaneously. No need to supply ATP, and it's just movement of substance from high concentration to low concentration. And you're like, wait a minute, isn't that the definition for diffusion? It's true, right? Diffusion is an example of passive transport. As you will see in a bit, there are other examples of passive transport that allows material to move across the cell membrane. On the other hand, we have active transport, okay, and that requires ATP. Right? The movement is going in the opposite direction. It's going from low concentration to high concentration. The best way to think about this is rolling a ball downhill. That requires no effort. It's going down the concentration gradient. That's passive. Okay. On the other hand, if you want to roll the ball back up, it's going to require energy. Right? It's going from an area that's low to an area that's high against the concentration gradient. That's why it's tough. That's why you need to supply ATP to do so. Okay? So two types. Depends on whether you need energy or not. Depends on whether you're going from high to low or low to high. Let's begin with passive transport. Simple diffusion, the one that we were talking about earlier, right? With the blue dye, right? With the Febreze, if you remember. Small molecules like water, okay, gases and hydrophobic ones, they are able to cross the cell membrane on their own with simple diffusion. If they're small enough, they can squeeze through tiny gaps in the cell membrane. And if they're hydrophobic, they would just blend right in. Because if you remember, the phospholipid bilayer has a thick hydrophobic core, right, in the middle that's made up of the tails. So anything that's hydrophobic will have no trouble blending in and then crossing the membrane. Okay? Like I said several times already, this process happens spontaneously, no energy is required. Osmosis is diffusion of water, right? So by extension, that is also a kind of diffusion, but we're just focusing on water molecule in that case, and that also requires no energy. Okay, so all those three scenarios that we talk about, the hypertonic, hypotonic stuff, water will just move on its own. Okay, it's a spontaneous process. Sometimes the molecule that needs to cross the cell membranes are too big, okay, too big, or they are charged. Right? If it's too big, it can't squeeze through the membrane, okay? And if it's charged, it won't be able to pass through the hydrophobic tails, right? Remember, charged particles are hydrophilic, but then the tails are hydrophobic. They don't mix, right? They're like oil and water, right? Not a good mix. So in these cases, in order to cross the cell membrane, they need a little bit of help. Okay, they need the help of a transport protein. Okay, so you remember from a previous discussion that some proteins are integral proteins, right? They spans across the entire phospholipid bilayer. And when you cross the cell membrane with the help of a protein, that is what we call facilitated diffusion. Okay, so simple diffusion, it just happens on its own, no help at all. Facilitated diffusion needs the help of a transport protein. All right, no energy is required, but you need the help of a protein. Two types of facilitated diffusion. Okay, the first one is called channel protein. In this case, the protein is an integral protein. Right? This is the outside, right? The ECF, the outside. This is the cytoplasm. These yellow molecules, they are ions. Ions means they're charged, right? Let's give them positive charge or something. So if they're charged, you cannot cross the phospholipid bilayer. The hydrophobic tail, the thick hydrophobic region here will block them. So the transport protein here, the channel protein, will provide a tunnel, a corridor for these charged particles to go through. Okay, so the tunnel will shield the ions from the hydrophobic environment. They're able to pass through. Now, in this case, they're coming in because they're going from high concentration to low concentration, right? If the situation switches, if it's high on the inside and low on the inside now, then they would just go outside. The channel protein does not dictate the direction of movements. They just provide a means for the things to pass through. Okay, so no energy is required. They are only using the protein because they cannot do so on their own. That's why it's called facilitated. Okay, someone has to help them. That's what facilitation means, right? To facilitate someone is to help someone, right? The other kind of protein that will help with the movement is called carrier protein, right? Channel protein is a tunnel. Carrier protein is more like a revolving door, you know, at the mall, right? Like you can go in and you push yourself and then you exit on the other side. The molecule will hop on into the carrier protein. Okay, it will turn and now it opens on the other side and spit it out. Okay, and it could keep on doing it. In this case, again, there's no ATP requirement. They are still going from high concentration to low concentration. Okay, but the big difference is that over here, the protein doesn't move. The protein sits there with a tunnel in the middle. This one, the protein actually undergoes subtle changes in shape, right? Subtle change in shape that translocate, that moves the molecule across the membrane. Okay, so big molecules like glucose would use a carrier protein. Charged particles would use a channel protein. Okay. So these proteins will affect, you know, the permeability of the membrane. Sometimes the composition, as we learn, also affects the permeability, right? How much saturated fatty acid is in the membrane, how much cholesterol is in there. All these would affect the fluidity, therefore the permeability of the membrane, right? The amount of proteins that are in the membrane will also affect the permeability. More proteins will give it more structural support, right? And it will lower the membrane permeability, for example. So there are other factors that would affect permeability, right? Temperature, pH, ionic concentration, right? What kind of environment they're in. And in the beetroot lab, you will be looking at the effect of temperature on the membrane permeability, right? Does it increase the membrane permeability? Does it decrease the permeability, right? We use beetroot because beetroot has a very prominent color to it. It's very easy to see. The colors are retained within the cell, but if the membrane is disrupted, if the membrane increases its permeability, then those pigments starts to come out, right? I don't know if you guys make soup with beetroot before. Anybody try to make soup with beetroot maybe? Otherwise, like this would not be a good analogy, no? Now, if you did, boiled it, for example, in your soup. Then afterwards, you scoop it out. The whole beetroot is just colorless. Okay, it's transparent. All the color leaks out into the soup, right? So that's an example of how temperature is affecting the membrane permeability. Okay, so based on this description I'm giving you, you can think about what kind of hypothesis to write. Remember, you don't have to explain things in the hypothesis. You just have to state your position. Like the one on the quiz, right? I asked you to write a hypothesis for, testing whether driving with the windows down will increase fuel efficiency, right? So what you needed to say was that driving with the windows down will increase fuel efficiency. Or you can say it will not increase efficiency, right? So some of you started explaining, oh, because of the air conditioning and so on and so forth. That's not necessary. You just have to state your position. And also, some of you wrote this. Some of you wrote on the test. Driving with the windows down will affect the fuel efficiency. And you didn't get any marks for that. Why not? What's wrong with this hypothesis? Let me say it one more time. Driving with the windows down will affect the fuel efficiency. What's wrong with that?
I think it's because the wording, you said affect, you want to know if it will increase or decrease.
That's right. I want to know whether it's increase or decrease, right? So in that hypothesis, there's no mention of increase and or decrease. You just say it will affect, like how will it affect it? Does it make it more efficient or less efficient, right? You got to take a position, right, in your hypothesis. Okay, so when you write the hypothesis for the beetroot experiment, make sure you take that into consideration as well. Okay. So that's where I left off with my other class. I know, it's a little bit early, but I want to keep the class in the same place. So that's where we will end. So please watch the video, do as much of the lab as you can, and then come back on Tuesday. We'll take up some of the, we'll finish the rest of the lesson, and I will help you with some of the questions in the lab as well. All right. Thank you for coming. Have a fantastic weekend.
Sorry. Oh, sorry. I have a question.
Sure, go ahead. Yeah.
Like,